Hello and welcome to the stream. This is going to be a fun one today. We've got it's a little bit different. So we're not going to be designing DNA. Instead, we're going to be looking at some that has already been designed. We're going to be speaking with a good friend of mine, Gabriel Licina. He's a biologist who has worked on, frankly, just a lot of really cool stuff over the years. So we're going to be chatting with him. We're going to talk about some of the cool projects that he's working on right now. Probably a bunch of other sidetrack stuff because that's how we do. And yeah, so without further ado, I'm gonna let Gabriel introduce himself. So let me just get this switched over. That should be the thing. All right, Gabe, you should be good to go.
Cool. Hi. Oh, there I am. And I'm lagged. Great. Hi, my name is Gabriel Licina. I'm a self-employed biologist, I guess. I do a lot of projects basically, I'm a facilitator. I help people build labs. When people have a crazy idea, I try to help them with their crazy idea. And Justin is a great friend, and he was actually my roommate for a while, and we worked together on a lot of different projects. And so if at any point you were like, oh my God, Justin was doing some crazy stuff in a lab, that was actually probably my lab. So, this is going to be fun. We're going to talk about some new stuff now instead of the old stuff.
Yeah, I mean that's pretty accurate. A lot of the I mean, pretty much all of the most ridiculous bio stuff that's been on the channel has been in your lab. So like the meat berry, the neuron thing, the lactose thing. I mean, it was all filmed at one of your labs. So that's gonna be fun. Cool. So internet meet Gabriel. Very exciting. Now I'm gonna pop over to benchling view. Got some stuff open, so I gotta close a bunch of stuff. I've been doing a lot of benchling work over the last couple of days. So you know what? There's a close all other tabs thing, and that is what I want. Also, I just realized I did not actually upload the ENTs thing like I was supposed to, because I am a turnip.
Not the plasmid?
Yeah, no, I did all the bee stuff. I didn't do the end stuff. So while I'm doing this, do you want to? I'm gonna make your window a little bigger so they can see you better.
I don't know. It's so laggy.
Yeah,
I don't even have my hat on anymore.
There's a latency.
There we go.
There's a latency. So like there's a lag
Okay.
between the streams. So don't worry about that.
All right. So we're going to talk about bees. So I already saw in the comment section that people are like, "Oh, hey, make some bees that are resistant to pesticides." Well, yes, actually, that's what we're trying to do. So it turns out that the horizontal gene transfer in bee guts is really high. I mean, that's just fundamentally cool in and of itself. Like when the bacteria in the bee's guts have genes, there's like a lot of hot swapping going on. And so some researchers were like, hey, let's see if we can make these bees immune to or like more resistant to neonicotinoids, which is, as everybody is probably going to be comfortable agreeing, is part of colony collapse disorder. And so it turns out that you can do this. And so they picked a very, very common yes, fire is just oxidation sped up. I have to not look at the comments.
Yeah, don't
okay,
read the
so don't read the comments. Just
I'm
talk.
maintain the comments. You talk.
Okay. They found a very common gut bacteria that's maintained in lots and lots of bees, regardless of region. And then they put in their gene of interest. And then they fed the bacteria back to the bees. And it turns out that it works. The gene of interest got transferred from the bacteria into the bees. Like, no CRISPR necessary, no crazy hacking necessary. It's just like the bees do that naturally, which makes a lot of sense because most of our biological engineering technology comes from biology. So biology is actually way better at biohacking than we are. And so this is like consistently. And so this is actually fundamentally the sweetest hack because, the bee gut is doing the work. I don't even have to do the work.
Yep. That's one of those
Yeah.
things, like, I mean, you mentioned CRISPR. So like, this is one of those, like, even on this stream, like, I get comments constantly being like, what if you use CRISPR? What if you use CRISPR? It's like, CRISPR is not actually even a good tool. It's like I'm seeing people like,
It's
what
a tool.
it is a
It's
tool.
a tool.
Well, that's the thing. It's a tool, and I'm seeing people who are like,
Right.
What if we're like,
no tool is a great tool if it's applied to the wrong place.
right, exactly. And like, so, I'm seeing people who are like, what if we like CRISPR'd fucking COVID? And I'm like, that's not.
That sounds like a terrible fucking idea.
It sounds like a terrible fuck. He's like, what if we got rid of the ACE2 receptor? You'd fucking die. Like, you need that. Like,
yeah.
So yeah, that's a thing. But yeah, so bees. So I've got the three things that were in that folder. So is bee symbiote construct the thing that we wanna look at?
Yeah, we want to look at the bee symbiote construct.
Bee symbiote construct, which unfortunately does not, in fact, turn bees into venom.
Yeah.
Which is, I find, rather disappointing. But life is often disappointing. Why is this only showing up as linear map? That's really strange. Okay, so I'm moving your little window around. I'm going to define this as, is it a linear thing or is it circular?
It's going into bacteria, so it is circular.
It is circular? Okay, cool. For whatever reason,
Yeah.
when I imported it into Benchling, it registered as linear for some fucking reason. Cool. Well,
learning experiences.
Learning experience indeed. This is interesting. So so this is my first time seeing this plasmid. So I'm learning at the same time the viewers are.
So tell me what you see.
So the first thing I noticed is it says Varroa dsRNA. So is the neonicotinoid resistance from Varroa mites?
Yeah.
Interesting. Very, very interesting. Okay, cool. So I'm seeing a tubulin alpha. I mean, did Andreas make this?
Andreas definitely made this.
Yeah, this looks like
you can
Andreas
tell because he loves because he loves to smack in so many freaking resistance or cut sites that it's just like
okay. So
you're like, oh my god, any enzyme I use is just going to make this shatter.
Right. Okay, so for the viewers who don't know who Andreas is a friend of ours who is a brilliant genetic designer who lives in Austria, but. For those unaware, it is super duper illegal to do any sort of genetic engineering in Austria without like a literal mountain of paperwork. And even then, they'll still probably tell you no. So
this is where I come in.
Yeah. So what Andreas does is he designs plasmids, and oh boy, have there been so many plasmids designed by Andreas. And then he'll have them synthesized, and then someone like myself or Gabriel will then actually like do the bench work for it. And the reason we were kind of joking is Andreas is a genius, but he also has some very specific design habits.
Yeah, so you can actually look at the construct and be like, oh, that's an Andreas construct, which is not a bad thing. I'm not talking shit here. I'm just I'm like his signature's on it.
Yeah, right.
That's the thing.
Like,
I just
sign their name, he just adds a bazillion restriction enzymes.
Yeah, and
so I'm seeing a bunch of like.
I think I gave
tubulin stuff. What's the deal with all this tubulin stuff?
So I need to pull up the.
Okay. So I'm seeing baculoviral repeat. What's that?
Mm-hmm.
What's that about? Tell me about.
That's. Go ahead.
No, is it?
I lost the.
What part of the what's that do? Is this going to be done in baculovirus?
It's a subset of baculovirus.
Okay. So,
Yeah.
interesting. So, like, at some point, is this going to be loaded into a baculovirus, or is it like?
Yeah, and so what we have to do though is
Oh,
ATCC is the basically the company that allows you to buy stuff. Like there's kind of a monopoly on um, uh, getting a hold of cells, and so because there's just like one big company, and so what they've done is that they say that they have this bacteria, but they won't let you buy it. So the first step of this project, before we even get into the loading it in, the first step of this project is to take a bee and extract the gut bacteria and isolate. My voice is too high.
Yeah, I fixed it. I think. Just give it a second to update. All right, people in the comments, is that better? I mean, Gabe.
Yeah, everyone says better. Okay, so
okay, cool.
Cool. So what we're gonna do is we're gonna have to go full wet work. We're gonna have to dismantle a bee and then isolate the gut bacteria that we want to use using a very weird and odd and highly specific protocol for isolating this bacteria. But luckily, because it's weird and highly specific, it means that it actually has a greater chance of being successful, because the chances of it not working are high because it's biology, but the chances of it only working in the way that I want it to are also fairly good because it's really specific and weird.
Okay, so just to clarify, there's two parts to this project, right? So part number one is you're going to try and isolate this weird bacteria from bee guts that will allow you to put new DNA into the bees. And then part two is this piece of DNA, the thing that we're looking at on the screen right now, is that piece of DNA. So this piece of DNA would be put into the bacteria that you isolated from bee guts, and then you would feed new bees the modified bacteria, and it should end up with this bit of DNA in the bees, and make them neonicotinoid resistant. Is the
right?
gist.
And one of the coolest things about this is that one of the biggest issues with doing genetic modification is that the bacteria don't care about what you want. Like, they care about being successful. And you being successful and the bacteria being successful are not the same things. And so they're better at doing genetic engineering than we are. And so they'll just cut out genes that they don't want. Even in big industry or academia, this is an issue. So what I'm hoping will work here is that because the bees will survive more, that's going to be an evolutionary pressure to make the bacteria also want to maintain the modification. So it's actually a really slick hack. Yeah.
That's interesting. Because, I mean, the neonicotinoids should leave the bacteria alone. Like, it doesn't really affect them. So, considering that, then the only selection pressure would be, do bees not die? And if bees survive, then bacteria survives.
Then the mod works.
Right.
Yeah,
which, I mean, that's pretty slick.
I can't stop myself from looking at the comment section. So what are the negative pressures or the negative effects, the potential negative effects? Well, it turns out that we're kind of fucked in general, ecologically. And so, one, we should really just be trying a whole lot of stuff, because the alternative is just to lean into it and die. But two, who is going to argue about bees not dying? And what's the negative effects of bees not dying? I don't know. You get to eat an apple.
Yeah, I mean, so that
But
I see one potential
it's a
negative.
hard argue.
So the one potential negative effect of this is, would this gene spread to other species, or even, I mean, even the varroamites? Like, if the varroamites become neonic resistant after, nomming on the bees, does that mean that varroamite issues are going to be way more of a problem now? There's that. Or if other species are become neonic resistant because of this, how do you, like? I mean, that would very quickly make neonicotinoids useless, which I see as a win.
Yeah, this is just a win. This is a
Like,
win no matter
yeah,
how it
basically,
plays out, as far as I'm
if this
concerned.
plays out, it basically means you can't use neonicotinoids, which I'm like, yeah, but that's good. We need to not use neonicotinoids. Neonicotinoids are fucking awful. So that sounds like a win. So
Yeah,
basically, like, our
and
three
I'm
options are
definitely
like,
not going to.
our three options are basically like, more healthy bees, no more neonicotinoids, or healthy bees and no more neonicotinoids. Like, either it either forces the industry to stop using the thing, or it makes the industry stop using the thing. Like, this seems like all win to me.
Yeah, and I mean, um, like there's definitely been
Dude, this
ramifications
is, wait,
for
sorry. I
GMOs.
saw a very funny comment. When you grow plants with proper biology and trace
Trace
mineral,
mineral. Yeah, yeah,
you
yeah.
don't need
Hey,
pesticides.
hey.
That's not how that works. That's not how anything works. Dude,
Yeah. So there's
healthy
definitely
plants is
been
just better
negative.
food for all the things that want to eat them.
Yeah. So there's definitely been negative ramifications for GMOs being used, but you have to understand that it's a technology. And so I can have a hammer and I can hit you in the head with the hammer. And we should probably not do that, right? But I can also build a house with a hammer. So part of what we need to grasp here is that how we use our tools is an us issue. It's not a tool issue. Tools are great. We're like live streaming with microphones and headphones on computers, and you guys are able to chat with us in the chat window in real time because of all of these tools. So we like tools. We just don't like the way that some of them have been used. So when we're thinking about genetic modification, we need to understand and be cool with the fact that some people have improperly used the tools, but it doesn't mean that the tool is bad.
Yeah, I mean, I think that's really the takeaway, and that's sort of the point of these streams, right? Like, it's just to, and the channel and all that kind of stuff. It's to show that genetic engineering, while it has been used poorly in the past, and even then it hasn't really been used that poorly. Like the biggest genetic modification that people are aware of is, I mean, there's like the BT crops, and there's the Roundup Ready crops, right? And like Roundup Ready crops, take a lot of slack, but or to take a lot of flack from people, but like, that was a really clever mod, and it's led to huge amounts of increase, like a vastly increased ability to produce food at scale, which last I checked, people enjoy eating food. And yes, there's an argument to be said of like, we wouldn't eat it if we wasted less food, but humans suck, so good luck with that. And then the BT crops are fantastic. Like, BT crops mean we need to spray less pesticides, and like they're completely harmless to humans. So like, that's already great. I don't know why they get such bad press. Like, BT crops are great, but then you get into the like, people get all fucking weird about genetic engineering, where you're changing like one gene, right? Like, you're, in this case, like, you're making the bees neonic resistant. But like, people are totally fine with it when you do nuclear gardening and you're changing like that, or like, you just throw
Oh
mutagens at your
yeah,
plants, and like,
atomic
dude,
gardening.
I want to try.
Or we could go back to the old school method of genetically modifying animals, and you force two dogs to fuck, and then you take the dogs you don't like and you put them in a bag and throw them in a river. So I mean, if you don't like GMOs, why the fuck do you hate puppies? Like,
Yeah,
what's
right.
wrong with you people?
So we got just quickly, we got a somebody kicked in, so I just want to say thanks. So I can't pronounce your name. I'm
mhm.
sorry. It's Jauhini. I'm sorry, I can't pronounce your name. I suck. But so he said, or they say, there's been talk of modifying various species to produce useful materials for humans. Um, are there any interesting ones bees could be modified to produce? I don't know, man. Like bees produce a lot of things that are already pretty useful to humans, like wax and
Yeah,
honey
but and also
and
also
food.
there's this the concept of what you got here where they said metal producing plants. Okay, phytoremediation is totally a thing.
Dude,
Like people take sun.
there's one species that
So
does phytomining so hard that it bleeds nickel. Like if you poke it, like just green shit comes out, and it's just like nickel concentrate.
yeah,
It's
yeah,
fucking
and
wild.
so like you have to understand the whole infrastructural chain, if you want to put it that way, or ecological chain of how these things work. And it's like, we don't need to modify the bees. I mean, the alternative, so this whole modification is because we've already done something wrong. In my perfect world, like, we don't have to modify the bees because we're not doing stupid stuff with pesticides and agriculture. But our entire agriculture system is kind of broken right now. And so, this is like a Hail Mary pass to get a fix because I'm not going to fix agriculture by just going vegan organic. It's just not going to fucking happen.
Yeah, also
And
like
so
the other thing with that one is people are like just go vegan, it'll save the world. Okay, first of all, fucking no. Second of all, like
let's no
and then
no no
it just gets
no
crazy
no
and like
don't thank the vegans. That was my fault. Don't
no,
do
I
it.
it's so frustrating. Like anyways. We got a question. Have you worked with any type of radiotrophic fungus yet? No, but a friend of mine
We
has.
did have the blood fungus.
Okay, we don't talk about that. we
No,
don't,
okay. We
no.
just
Okay.
we
Okay.
just burnt it. We're not gonna.
Um,
Okay.
so the way that the radiotrophic fungus. So for those who aren't aware, radiotrophic fungus are a type of fungus they found growing in the fucking reactor core of Chernobyl. That was eating the radiation coming off of the waste and growing off of it. And so they actually figured out how this works, and it's through the melanin that the fungi produce, and they use it as if it's like think chlorophyll but black. So it absorbs the radiation and turns it into fuel. And so there was actually a paper by I want to say it was the NIH where they modified bacteria to produce melanin, and they grew like way better when you expose them to UV light. So a friend of ours has actually produced a plasmid that makes bacteria produce melanin that he's going to share with me at some point. So eventually, I'm gonna have. So we're actually gonna do this test. We're gonna basically get 2 plates of bacteria, and one of them's gonna be modified with melanin, and they're gonna be growing in the same media, but one of them's gonna have like intense UV light on them, and we're gonna see which one grows faster, and see if we can actually do a little bit of like radiotrophic, like growth, which just sounds wild. Okay, wow. Let's just take a wee break to just ban some people. Aren't comment sections so much fun?
I'm having a great time.
No,
I love conflict.
Yeah,
That's
no,
how
it's just,
that's how things grow.
it says, are the radiotrophs absorbing the gamma rays or the beta particles? Nobody knows. There's not been enough study on them. Because the one problem with the radiotrophic fungus is they're also like super duper pathogenic and very angry. Unsurprisingly, thing that grew up in the core of Chernobyl is not a happy organism. So working with it's really dangerous. Which is why people made the synthetic version of like, let's just modify some E. coli. So yeah, that's it. There's not a lot of info about it because it's not very well studied. Because A, getting a sample is really hard. C, you need to go into the core of Chernobyl, and working with it's really hard because you need like a BS3 lab just to like grow some, and it's awful. Please.
Don't feed the trolls. Don't feed the trolls.
I wasn't going to. I mean, someone's asking to be banned.
Okay.
Okay. I can.
Yeah,
I
and
can.
asking to be banned.
I
I
can.
mean, like,
I can
does he
grant.
want to be banned?
I mean,
Or yeah.
you know,
Um.
Alakazam, your wish has been granted. Fucking like, bye.
Yeah. Okay. So so let's get back on topic here.
Yes.
We were
Oh,
talking
so
about
I'm looking at this. I'm looking at this
bees.
plasmid, right? And this. I mean, it looks like an Andreas plasmid. I'm seeing a lot of stuff that I have no idea why it's here or what it does. Because the only thing that to me is necessary is the like Varroa bit and the baculovirus thing. And for the life of me, I
Mhm.
can't see where did the Varroa thing go. So I'm seeing like RNA polymerase subunits. I don't know why those are there. I'm seeing this tubulin alpha. I don't know what that's for.
So part of the Andreas method. So one, Andreas is, like I said, far superior to me in
Right.
design. And so part of the Andreas method is he'll just go into the research that already previously exists, and he will take what he needs from it and he will smack it in there. Um, the way that, like, I mean, you know that there's like a U pipe underneath your sink, right?
Right.
Yeah. And you know why there's a U pipe underneath your sink.
Okay.
And but you don't necessarily need to know why the U pipe is underneath the sink. This is also like, actually, I'm going to try to segue here. This is how we ended up dealing with like agrobacterium.
Mm-hmm.
So like we use agrobacterium for modifying plants. This is like our primary method for modifying plants. And you have all probably seen agrobacterium in the wild. You just don't know it. It's like when the trees get the weird little knots on them.
Yeah,
Like,
we've talked
heck is
about this
that?
on previous streams. I'm gonna pull this
Yeah,
up.
and so
I'm gonna pull
yeah,
up a
and
picture.
so one of the things with the agrobacterium is because it's biology, we don't actually 100% understand how it works. And so we just remove stuff until it stops working. And then we're like, oh, that must have been important. As opposed to a computer where we like put it together a code where like everything is fundamentally laid out because we built it.
Yeah.
Biology is not our tech.
Yeah.
And so with the agrobacterium, they made the most streamlined, simplified type of agrobacterium by just taking pieces out. They're like, does it stop working? Okay, you can't take that piece out. You take a piece out. Did it stop working? No, you can't. Yeah, that thing. And so,
Yeah. So just quickly, the thing we're
so
looking at
part
on
of
screen is what agrobacterium does naturally. Like agrobacterium naturally genetically modifies whatever plant it gets in, basically. Um,
and it's
and
everywhere.
it's everywhere.
It's
Anytime you've
everywhere.
seen these like weird like tree tumor looking things, like that's what that is. So basically,
It doesn't really hurt the tree.
yeah, no, I mean, because trees don't care. Like cancer does not affect trees in the way that it does like mammals. Like tree just goes like, oh, whatever. There's a lump. Who cares? Um, and I mean, like you've almost definitely seen these before. Like there's all kinds of different ways that this can look, but I mean, it all looks like weird like galls, basically.
Yeah, they're just yeah. No, it's not a parasite. It's co-evolved.
I mean, it's a little bit of a parasite.
Uh,
The tree does not like the tree doesn't benefit from this in any way. It's just a tumor. It's like
but it's no, it's
but
but it's not a tumor. It's just that we can't talk about plants like they're people. Like it's not a tumor. It's
I mean
a
it
different thing.
it
The
is
whole conversation is anthropomorphization right now, and it just doesn't.
but I mean
No.
it is a tumor. It's an uncontrolled growth of cells. It just happens to be infectious, which I
It's
mean
not uncontrolled. The bacteria are controlling it.
well, I mean, yeah, but the tree isn't. Like that's like saying I've got HPV. It's only cervical cancer. It's not control. It's not controlled. The virus is controlling it. It's like, well,
It's different. It's different. Plants are fundamentally different than us.
plants are fundamentally
Like
yes, that's fine. But like it's one of the, if it looks like a tumor and it acts like a tumor and it tastes like a tumor, why are you eating tumors? But like, um, you know,
delicious tumors.
we had another question with regards
Yeah.
to plasmid retention problem. Have you considered using an integrative
Mhm.
plasmid? Does this plasmid integrate? Does it have the ability to do that?
This plasmid actually doesn't have an integration site, if I remember correctly, though Andreas does like to fiddle with stuff.
Right.
But but it doesn't matter. It doesn't need to have an integration site because every piece can always be edited out. And so directed evolution is actually one of the coolest techniques that we've ever worked on for doing genetic engineering. And so, because it sticks, because it matters to them.
Fair.
So, does that answer the question?
I think so.
Yeah.
So I mean, it's an interesting plasmid.
I feel like it's a more I think it's a more elegant solution to not have integration.
Right. The interesting thing about this is, so I'm looking at this, and the problem is I don't know how neonicotinoids, like I'm familiar with neonicotinoids, but not their specific mode of action. So I feel like if I knew, in fact, here, let's just look this up. Is it gonna
click Google
mode of action? Okay, so like I want to see what the mode of action is. Let's see if I can just grab a graphic real quick. Oh, interesting. Okay, so it messes with acetylcholine. That is not what I would have guessed. Okay. So it's acetylcholine esterase, cannot break down neonicotinoids. And so the neonicotinoid presumably clogs up a receptor. Yeah, so that was not what I wanted. Cool. So it messes with receptors. So let's look back at the plasmid and let's see if we see anything that is obvious. See V-type proton ATPase, or sodium potassium transport ATPase. So I think what I'm looking at is basically a collection of different parts that essentially force the neurons to continue functioning even when neonicotinoids are present. Like if I were designing this, I would have assumed that the obvious solution would be like something that breaks down neonicotinoids, but I guess the easier solution is just
but that's
make
all
them useless.
That's an entire enzymatic pathway, you know.
Yeah, but I mean, you can just break how far down you got to break them before they stop working. Like if you only got to like knock off a functional group, that could be one enzyme. Like kanamycin resistance or like ampicillin resistance basically just breaks the antibiotic at the little square thing.
You're spooling right now.
What?
We glitched for a second.
Oh yeah, but I don't know what.
We're back. We're back.
I don't know what the deal with my internet is, but it's a little shite. It's a little bit shite, and I don't know why. And it's been like this. It's been like this for weeks, and I've called and I've yelled at the. I say yelled. I've expressed my frustration to the internet company, and their response was basically like, shrug. Like, everyone's using the internet. I don't know what you want from me. I'm like, oh, okay. Well, I'd like you to fix it. Anyway, whatchamacallit? So yeah, I'm surprised that just break down the neonicotinoids isn't the obvious solution, and instead the solution is rework every single sodium potassium transport in the entire thing.
Yeah. Well, my assumption here, like based off of doing decades of research, is they probably already tried to do that, and something very bad happened.
Yeah, I mean that's
and
usually
then they
how
were
that
like,
goes.
They were like, "Oops, gotta tap out now. We're gonna do something different."
Yeah. So I saw a question go by, which is why aren't we using CRISPR? And I think we've already addressed this, wherein CRISPR is a tool but not magic. Like people are like just CRISPR it. Like CRISPR's a protein. You can't just like spray a bee with CRISPR protein and have it like do something. That's not how it works. You have to get the protein into the cells, which in this case,
you still need a vector.
So you still need a way to get the thing into the cells. So it still needs to be on a plasmid, and you still need a way of getting it into the bees, which traditionally, the way that you modify insects, because people are like, before this really clever use some magic bacteria to like hot swap it, the way that you modify insects is you get a bunch of like insect eggs and a very tiny needle, and you do micro injections. So you literally just like boop the DNA or the straight up proteins into the eggs, which is insane. And
Hello, little insects. I am your mama, and now I
yeah,
will boop you with cheese.
I saw
Um,
a paper on the first genetically modified ants. And what they did, and I was like, okay, so they must have made a plasmid or something. No, they literally got like several thousand ant eggs and some poor fucking grad student who had to sit there micro injecting not even DNA, CRISPR proteins. Like they actually just injected straight up Cas proteins and a guide RNA into the eggs. And it was like, the study was like, and we did this to 3,612 eggs. And I'm like, that poor fucking grad student, that's so much work. Like,
yeah.
that is so much work.
Poke.
Yeah.
Jesus.
Yeah, dude. So I'm gonna pull up an image of what a micro injection system looks like.
Actually, we got some people looking for more details. Can
Okay.
you bring up some papers?
Sure. In just a second. I wanna see
Yeah.
if there's
You just want to share eggs getting stabbed.
yeah. I mean, it looks exactly like what you'd think. Like it's literally a tiny needle, and the way you make the needles, like you don't buy them usually. Like normally you just get like basically a glass pipette, and you just heat up the end with a torch and just pull it really thin. So you get like, this whisper thin needle, and then you've got to get like it's a mount that you mount the thing in that lets you like have little dials to adjust the thing. Think of like basically like controlling like a milling machine, except it's a tiny needle and some very, very sad ants.
Yeah.
In this case, they're saying, what are the ants supposed to do? How were they modified? They just knocked out a gene. So I think it was a
Again,
it was like a sensory
you gotta
receptor gene. So
yeah, you got to remember we don't know how things work most of the time.
Yeah.
And so most of our bioengineering is selectively breaking things and then being like, oh, so that's what that's good for.
Yeah, basically. So like in this,
basically
it was like
they broke a lot of ants.
Yeah, they like turned off a hormone receptor and was like, I wonder what they do. Whereas like I wanted to make like ants taste like bananas or whatever, and they're just like, let's just make them broken a little. I was like, that's not
Okay, let's get that. We've got multiple asks for papers. Let's get those papers up.
I mean, I got this one. So engineered symbiote activity honeybee. This thing is this the paper you wanted?
Yeah, that's the paper.
Okay, so yeah, this is the paper this came from, and that is a picture of a dissected bee, isn't it? Let's see, D photograph of dissected bee. Okay, Jesus. Okay, cool. So this is the paper. By the looks of it, F is stained. Okay, cool. So what they did was they cut the bee open. This is presumably its bits. And then they took some fluorescent images of the inside of the bee stomach. And the glowy seems to be the weird species of bacteria that you need. No. So I misread this. So on the left, E is the natural normal bee gut. On the right is the modified gut bacteria fluorescing. So they added a fluorescent protein to theirs as presumably a reporter of some kind, just to prove that they could, A, modify the thing, and B, put it into a bee and have it survive, which evidently it does. So that's pretty fucking great. So that's neat. So the bacteria is modifiable, so that's exciting. And yeah, here we go. PDS GFP. So they use GFP as the reporter, which would explain the fluorescence. This is very cool. So like in this case, they did symbiote produced RNA interference can improve honeybee survival after viral injection. So is it both? Like,
yeah.
So do you hit them with a virus?
That's just one paper. There's a whole thing. Yeah.
Okay.
We're making super bees here.
Weird. Very weird.
Uh-huh.
So this is the yeah. In this case, they did the varroamite. Um, it's just RNA interference. That's weird. It's weird and interesting. I mean, RNA interference is sort of the like the easy cop out of like not even easy cop out. It's a specific tool, but it's also a very good way. Like it's a good thing to start with, rather than like trying to express a gene. It's like let's just see if we can turn one off. Because I mean, just like we said before, like we don't know shit, but like biologists don't actually know shit. Like
Oh
we're
yeah, you get
we're
every
just
biologist that knows everything about, let's just say, bees into one room, and you're gonna have maybe 2% of understanding how shit works.
on a good
That's
day.
just a
Like that's
fundamental on a good day. And this is so hard. I saw a couple of people mentioning that they were coders. This is so hard for coders to deal
it
with
really is.
because they're
It
like,
really
yeah, you
really
do the
is.
this and then the this and then the this and then the this. You got your Legos and you snap them together. And it's like, we've got a big handful of Jello and I don't even know if I should put it in my mouth. It's like really like this is being a biologist. And the thing is that they're alive. Your software is not alive. Your computer isn't alive. If you
Yeah.
shut down and air gap your computer, it's off,
No.
right? But if I ignore the bacteria in my lab, they will just keep doing whatever the fuck they want.
Yeah.
And they'll just like, they have independent desires. Um,
I mean,
and
we talked about this on an earlier stream where it's like biology is like coding in the sense that you like make some code. The only difference is the compiler is actively hostile and like explicitly does not want to do what you want it to do. So it will go out of its way to fuck your shit up. Like imagine if you were like trying to code like you're like I'd like to make a website and like every now and again the website like the thing would just close like flip you the bird and then like switch to doing something totally different. Like you're like, I'd like to make a website for my photography. And then like just one day it just decides that nope, today we're doing porn. And it's just like, what? No, why? Like that's not what I wanted. And
yeah,
like it's just its desires and your desires are diametrically opposed. Um so getting anything to cooperate is real hard. Like this I mean something I learned from Gabe is that if you don't need to do the wet work of like building a plasmid, for example, don't. So this is the sort
yeah,
of thing that like
yeah.
you just pay a
Just to
company to just
fucking,
make this because it's too goddamn frustrating to
just,
try and do it
yeah.
yourself. Um,
Yeah, everybody wants to be that lone hacker cowboy that's doing the snippy snips,
when it's
all
like
the
it's
different
a
snips.
huge waste of time if you can avoid it. Like, and you end up
Yeah,
saving like you also end up saving money. Like it sounds expensive when you're like I paid $1,000 to have this made, but like $1,000 to have a thing done, that's like days and days and days of your life saved that you don't need to like sit at a bench wondering like why is a thing broken. Like that's worth every penny. And also like then you don't need to buy the restriction enzymes and all the other enzymes that you need to do all the bench work. And you don't need to like run 4-hour PCR reactions constantly. And like if a reaction doesn't work and it takes 4 hours to run, guess what you get to do 400 times until it works. And like that burns days and days and days and days. Like literally in the other room, I've got a PCR reaction going for the Silk project, and it's literally going to take 4.5 hours because I'm PCRing the plasmid that I'm working from has no useful restriction sites. So like you can see, and I'm gonna move your little window so that people can see this better. But
yeah.
actually, you know what? I can just open this up really quick, not to detract from your projects, but just to make this
No, 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 it's
obvious.
fine. Mhm.
So this is the and I like doing little spider silk updates because people constantly ask me about it. So it's nice to be able to just like show it off. So we're looking for V4. Yeah, here we are. So this is the latest version of the spider silk. Um, and this is the plasmid we're working from. So yeah, this is the backbone plasmid that I want to put the silk into, and it's nice because it's got an ORI for yeast, and it's got kanamycin resistance, and it's got the TEF1 promoter, which I really like. I like all of these things. However, you'll know.
Wait. Now I have thoughts and feelings about these. So the problem is that if the PI doesn't want to pay, then there's nothing you can do. That's why we don't work for academia.
Yeah, seriously. This is like why
I am the look at me. I am the PI now.
yeah, this is why we do things the way we do. Like people, I get asked sometimes, like, why don't you work with academia? It's like, because I don't want their fucking opinions. I just want to be able to do my thing. Like like if I find
somebody just mentioned the ENS project, so
yeah, we're getting there.
We got the NTAX. That was the next thing we were going to talk about.
Yeah, we're gonna get there. I want to just show this really quickly. So, if this plasmid would be useful, right here, literally where this cursor is, right here, there would be a whole big long section full of restriction enzymes that makes it easy to cut this open. Except as you may notice, it's kind of barren. So there's nothing there. There's no easy way to cut this plasmid open. So what I'm doing is I'm PCRing literally from this spot, like all the way back around to here. So this whole thing is one massive PCR reaction, just so that I have an open plasmid where I can stick a new thing in here. And so that will end up, assuming that works, I'm then going to cut and not even PCR. I'm never PCRing my silk gene ever again because it's awful. I'm going to actually, the PCR primers that I made actually have special little, like I've added restriction sites. So that way I can just take this whole chunk, which is my silk gene, and just shove it in here and make a new silk plasmid. So this way I can actually put my silk into an actually useful species of yeast instead of the garbage nonsense that is Pichia. So this will work in regular brewer's yeast, so I can actually make spider beer. And this I can just transform this directly. I don't need to cut it. I don't need to do any weird shit to it. I just take DNA, put in yeast, happy, like is how it should go. And if it doesn't, then I'm going to pay a company to do it for me because my ability to like put up with bench work is
is
non-existent.
That's what I'm for. That's what I'm for. I'm the bench work bitch.
People are saying it's too long for PCR. You just haven't tried hard enough. No, I have Q5. So the enzyme I'm using is called Q5. And so it can comfortably copy 10,000 base pairs of DNA before it starts making mistakes. It can do up to like 20, but then it'll make a lot of mistakes. So this is actually well within its capability. It's just quite large. So it's not an easy thing to PCR. So this is why the PCR takes so long. Like I'm running it for the first time now. I have no idea if it's going to work. It's going to go for 4.5 hours because the extension step is like 4 minutes. And then like I'm running everything at like max. I'm doing touchdown gradient. I'm like giving this thing all of the opportunities possible to work. And like I've gotten very comfortable with this PCR machine and this enzyme, and like I know it inside and out at this point just because I've fucking used it so many times.
Justin?
Oh, it's going to be garbage and it's not going to work. I'm entirely sure it's not going to work. No, past a point, if this doesn't work, again, I'm just going to send all of these pieces of DNA to a company and be like, you fucking do this. I'm not doing this anymore. And like,
yeah,
if that's the thing that I've got to like convey to people, that like, the how-to biology, you just pay other people. Like,
okay, but that's how you biology. That's not a real lesson about how to biology.
No, but it's how you biology too.
Learn.
It's
you gotta learn how to
call up the company. Suffer. You call up the company and you're like, please make me this DNA. That's how you go. That's how you do like. The problem is people get stuck on the building the DNA shit, and then like, that only at the very end they work on, like, does it work? Does it do the thing we want? Can we get it into the organism? No, fuck that. Cut out the whole first half of that, have a company make your DNA for you, and then just do the thing. And then, like, you can focus on, like, why isn't it working? Rather than, like, why can't I put it together? It's Lego. It should just stick. Like, It's very frustrating.
But the yeast still don't care about you, Justin.
But that's the thing, and the yeast still don't care. And like you have to grow them on poison just to get them to do the thing that you want. You're like, God damn it, you had one job. Oh my.
Okay,
So
let's
yeah,
talk about.
let's talk about some trees. So your other project. So you've got the bee thing is very very cool, and I'm excited about it. Over the next little while, I mean it's bee season, so I hear you're going to be cutting open some bees soon. You know, we'll get some tiny tombstones for the corpses, but like, you know, cut open some cut out. Oh my God, he's gonna have a meltdown. But anyway, so the plan is over the next couple of months you're gonna cut open some bees, try and extract the weird bacteria, and then once you got the weird bacteria out, you're gonna get this. We had it open. It's this one. So then once you've done the once you've got the weird bacteria, you're gonna have this made, and then you're gonna put it into some bacteria, and then put it into some bees, and so that's pretty fucking cool.
Yeah.
So yeah, I'm thinking that I mean this is a really cool project. It's a project that I'm big fan. So potentially once you've got the bacteria and you're like ready to make some DNA.
Right, using that highly specific, super weird protocol that somebody wrote like 3 papers about.
Yeah.
So like really great isolation, I think, actually.
Yeah. So the isolation the isolation's like 2 antibiotics and a weird media, right?
And it needs to be in a elevated CO2 environment because you got to remember the inside of a bee gut has less exposure to oxygen than the skin of a bee, so you got to use like a mammalian cell incubator basically. So it's not just the antibiotics, it's the antibiotics and the gas levels and the media.
So I mean it sounds like basically you know blend some bees, put it on the media, and in theory it should just do the thing, which sounds pretty weird and pretty cool. I'm excited to see how that goes. But yeah, assuming that works, we may end up doing maybe we'll do a little fundraiser, see if we can help get this construct made. Because I mean, neonicotinoid resistant bees sounds dope, and I'd love to see that happen. And we'll do some video stuff on it. So I think that'll be.
And the construct
cool.
is in terms of constructs that we've tried to make or have printed for us. The construct isn't super expensive. I'm just hell's a broke. Because I'm a random guy that likes to do biohacking as opposed to somebody that
is expensive,
capable of holding a real job. And bio is expensive.
Bio is so expensive. Oh my God.
Yeah.
Okay. Cool. So that's bees. Bees are dope. Now we're going to talk about some trees.
So we got bees. Now we got trees.
All right. Should I pull up the original paper? And we want to talk about that first?
Yeah, the PXY CLE41 paper.
Cool. So on a previous stream, I mentioned that there was a paper that was written and a patent for making trees grow twice the size in half the time. And this is the paper. It's called "Wood Formation in Trees is Increased by Manipulating PXY Regulated Cell Division," which that just rolls off the tongue. It's not like that paper, Fantastic Yeasts and Where to Find Them, which I love that paper. Not even because it's a great paper, just because it's a fucking amazing title. Anyway, so basically the TLDR is if you mess with these genes, and you have to mess with both of them.
So there's 2 genes. There's the PXY gene, and then there's the CLE gene. And if you mess with one, it creates lots of cells, but you need to have the second gene as a regulatory process to arrange the cells.
Yeah.
So it's like, again, we're going to go back and talk about Legos. You can grab a handful of Legos, but you still need to take the time to snap the Legos together. Or you can grab the Legos one by one. And so you need to actually have both of these things simultaneously upregulated so that you don't just get like weird sponge tree.
Yeah. And so I've got a picture up of what happens if you mess with the individual genes. So one of them you get, like, if you mess with just CLE41, you get like tiny trees. Like they're not happy. Because that's getting. I think the CLE makes more cells, whereas PXY is the one that actually does the distribution. I don't remember exactly the specifics of that. But
Yeah, no. So PXY does the distribution, and CLE does the snapping the shit together.
yeah. So basically, if you've only got one, you get tiny sad plant. If you got only the other, you've got weird stretchy sad plant. But if you've got both of them, they get like big. And
Big.
big. And so it's also increased wood formation, right? Like it's more lignin. Is that the?
Well, so so there's more research that's been done after this paper, and so my particular opinion is that saying that it just does this is
I mean, it hits a whole
a
cascade.
little super reduction. Yeah, it's a whole cascade because wood formation, I'm making quote marks, you're just going to have to catch up on the video. Wood formation actually means something completely different when you're talking about plant biology. And so this also means the diameter of the leaves. You make super chonk trees with this,
Right.
and everything is chonk. It's basically like
And this was just the first paper too.
And then this is just the first paper.
There was like they did a whole bunch. So one of the other things in later papers that I remember just from reading up on this was, so in this you'll see that it says 35S. So that means they're using the 35S promoter, which is basically like run this gene all the time in every single cell, which is not actually what you want. So one of the things that's in the patent that came from this is not only do you need to turn these two genes on, you have to use very specific promoters that only turn on in specific parts of the plant. And when you do this, then you go from like these weird, like derpy looking trees to just massive plants.
Right. And so here's where it's going to get weird. You guys aren't ready for this, but here we go. So I'm like, hey, why don't we take this cool gene hack that was, I'm going to say it, made by the lumber and sugarcane industry to increase their yields. And so, of course, it's never been used for anything else. And why don't we smash it up with permaculture and try to get some of that hippie nonsense out of the way as well, and use a planting method called the Miyawaki Method, which some of you may know is about high density multi-species planting. So this is not about increasing monocultures. This is not about getting higher yield. This is about actually just making an entire forest grow faster.
I'm trying to pull up a picture. Yeah, it's pretty dope. Like, I mean, this I love this project. It's just so cool. So the thing with this paper, and specifically the patent that came after it, or that came from it, or whatever, is it is the most airtight patent I have ever seen, ever. Like, not only are you not allowed to, you can't. Not only can you not use these genes and the promoters that go with them, in any of your stuff. Like, that's super patented. Like, they'll sue the shit out of you for it. They've also, the way the patent was written, it also covers anything else that messes with these genes. So, like, if you make like a new molecule of some sort that messes with these genes, it's still covered under their patent. Like, if you try and like mess with some other gene in another part of a pathway that like kick this, it's in their patent. Like, there's no way to actually touch these genes and not have the shit sued out of you, which is just the worst. So I'm pulling up a picture of the Miyawaki Method because the Miyawaki Method is dope. But it's basically the idea is you basically grow like little plants, and you just plant them really densely, like really densely.
Right, because so think about it this way. Like, we as people like to do things for optimization, but nature doesn't care about being optimized. Nature cares about being functional. And again, that's anthropomorphization. Sorry about that. But so this, like, when you see an acorn on the ground, you probably also see like another 100 acorns. So we're like, hey, we lose 70% of these little oak trees, so we should space them. The thing is that they have all co-evolved for density, right? We haven't even looked at how these, we just like threw our own model on it. We haven't thought about the fact that this is how the thing evolved. They co-evolved for density with a lot of other competing species. And it actually turns out that when you have high density multi-species planting, you get better plants as long as you're comfortable losing half of them.
Yeah. I mean,
so you just got to plant. So, so this is fundamentally different than how we do most planting, but it works. And this guy named Miyawaki had this paper in like 1965. And anytime you've seen that Facebook post where somebody's like, this guy in Pakistan turned the desert into a forest, it's because he was using that method.
Yeah.
And so that method is the baseline. That's how nature will function if you deal with it properly. If you stack genetic engineering on top of that, who knows what'll happen?
Super forest.
It'll be awesome.
Yeah, so
super forest.
So yeah, that's the thing. Like, if we've got all this land on Earth that's either been cleared, or even if it could have been cleared 100 years ago. Like, it's nothing. Like, it's not like the current generation's fault per se. It's just been fucked for 100 years. It's just the way it is. So the only way that I think humans are going to continue to exist on this planet is if we start going in and unfucking a lot of these areas, and most scientists would agree. So the Miyawaki method is a really, really great way of doing that, and I think the combination of this like well proven planting method combined with genetically modified plants, like there's people who get real weird about like releasing genetically modified plants, but I'm like 90% I think we're at 90% of species are extinct. Like of all macro.
Okay, let's rephrase this, because somebody is going to say something about this almost immediately, because that was not properly phrased.
It's not properly phrased, but like
80 to 90% of the species density has been reduced.
Yeah.
On the planet at this point in time.
And also like a huge amount of the macro species
lions
have gone
aren't
extinct.
extinct.
There's just a lot less of them.
And then, of course, and then there's that too. So extinction is a thing. We're just we've gotten really good at making that happen. And I don't know if it really needs help. So maybe we should help in the opposite direction.
Yeah. So, we're losing, so not only are we losing the, the mass, like we're literally like, even if the species haven't gone extinct, like there's just less of all of them. But we're also losing, just straight up losing species. Like, I mean, it's the difference between, like, if you were going on a road trip 20 years ago or 30 years ago, like, your windshield was covered in bugs. Whereas now, not a one. Like, the insect population is just fucked. Like, absolutely destroyed. Like, we're losing plant species.
And this is just in the last 50... This is the last 50 years. We're not talking about all the species that ever existed. We are explicitly talking about in the last 50 years, this is what happened.
Yeah. So personally, and of course this is just my personal opinion, I'm actually a huge, like, I like the idea of conservation. I just don't see it working. Because
conservation leans into the concept of pastoralism, as if there's a perfect state which we what are you conserving? Like, do you even there's efforts to this even know what's an invasive species?
Right. And you've got the efforts to save specific species, but it's never really been very effective. Trying to just work with one species never really works, because it's an ecosystem. Like, you need all of them, And so it's like a band-aid. Like trying to save one species out of an entire ecosystem will never really work, because if the ecosystem is still dying, like you adding a couple hundred more members of one species isn't going to fix the root problem of the whole ecosystem.
Let's talk about macro species. Let's talk about pandas. Everybody wants the pandas to fuck.
Dude, I would eat a panda so quickly. Like, I bet you they're they're delicious. I bet you they're like the most,
they're like the veal of the bear world.
They're right, like they're the most cared for animals on the planet. Like,
hey,
I bet you they're delicious, and as species go, like they deserve to be extinct. They went from predatory animals to eating nothing but the most like nutrient poor plants they could find, because it was the only thing that wouldn't run away. Like,
hey, I heard that species like niches, and so I gave that niche a species. So pandas, everybody likes to talk about saving pandas. Pandas are a front end. And so even if you save the panda, you genetically modify the panda, you put the panda back in the world, the panda is just the thing that you see that you're happy about, but it's not always about what makes you happy. And so the panda is the front end for bamboo forests and temperature and mountains and all of this other stuff. And so you can't just save a species. You have to do a fundamental rework of the system.
Yeah. So personally, I'm of the opinion that the whole thing is fucked. Like there's no going back to like the magical, wonderful diversity by just conserving really hard, right? Like just like cordoning off areas and being like, okay, we just don't touch these. Because I mean, first of all, people will always violate that because humans are bastards. And
we can't even stop violating social distancing while people are dying right now.
Yeah, so, seriously. We
we're like golden retrievers. We just jump. Yeah.
So there's that, but also there's the greater problem of like, okay, so you've cordoned off an area of like nobody touches this forest, but the temperatures are still going up and the weather patterns are still changing, so it's still gonna get fucked. So I'm of the opinion that like because that is the way that it is, and we're about to see even like higher levels of die off of all kinds of different things. Fuck it, let's just mix it up a little bit. Let's start genetically modifying things and just let it go.
Might as well try for something good,
right? Like let's make new biodiversity instead of
selfish little shits.
Right, like, and because that's the thing. Like everyone's like, that's an invasive species. It's only an invasive species because we noticed. Like in 1,000 years, it's just going to be a species that lives there. Like, so all these things that are like invasive species, yes, it messes with the ecosystem, and yes, it means that certain species will die out, and that's awful, and it's shitty, and blah, whatever. That's only
that was funny. The two pandas on the windshield. That was funny.
Yeah.
Okay.
So it's like I think the only way that is really gonna like what's in 1,000 years, it's gonna have found a new balance. Like maybe it won't even take 1,000 years. Nature bounces back very, very quickly as we've been seeing. Like humans fuck off for a month, and like Venice is full of fish and jellyfish and shit. Like it's been a month. Like that's 1 month. 100 years, 50 years, 10 years of humans not messing with a thing. And it just gets, I mean, like, look at Chernobyl. Like, Chernobyl is, like, lush greenery now. And yes, admittedly, some of the animals are a little bit fucked. But, like, it's a remarkably healthy ecosystem. Like, you see, like, this pond of fish that's, like, full of really happy fish that are, like, just chilling. Because there's no humans to mess with them. Like, so it's all this kind of shit where I personally, and, obviously, this is just my opinion, but personally, I see huge benefit to mixing up the biodiversity a little bit and, not necessarily throwing invasive species. Like, ideally, if you can use the species that are already there and diversify them through genetic modification or whatever else, like, this could be really interesting. It could make for interesting ecosystems. But I'm also a huge nerd, so I'm like, let's make glowfish. Let's just make all the fish glow.
Oh my god! Because
it's stupid and like it's fun. Like, because glowfish would never survive in the wild anyway. A thing that's fucking fluorescent is not very good at camouflage, unsurprisingly. Like, neon pink
Yeah.
is not a good color if you're trying to not get eaten. So, like, these things wouldn't persist
I
anyway.
mean,
Like,
unless you think you're a snack.
Yeah, well.
Sorry, I was,
What do you think of geoengineering? I mean, we're geoengineering real hard
We've been
right
doing
now.
geoengineering for like
About 200 years.
No. The Amazon rainforest, dude, Terra Preta.
Oh, yeah.
Like, geoengineering is something that we have been fundamentally doing for the entirety of human history.
This is true.
Like,
and also, people forget that, like, basically the entire planet has essentially or not essentially, it has been geoengineered. Like, we find, like, when you go through, like, a woodland in the UK, like, you're like, yes, these trees have been here for hundreds of... No, fucking, like, forests were tended areas. Like, if you were, like, hunting deer back in ye old medieval times in a forest, like, you don't want to be, like, bushwhacking through a bunch of low shit. Like, you go in and you clear out all of the low plants so that it's easier to move around and hunt some deer, because that's how you eat.
yeah, and
So,
the
like,
entirety of the Amazon rainforest that we're like so excited to protect was made through really intense, hardcore acts of geoengineering.
yeah, I mean, there's large tracts of it that, so it's not the entire Amazon that was done by humans. Part of the Amazon was actually terraformed by large mammals. So things like
macro species.
Yeah, so it's like the extinct giant sloths and stuff would go in and they'd eat things like avocados, and, like, or it's like there's all these big species that would eat plants and then spread them all over the place. Plus, you've got the human activity in the area.
I'm compelled to actually pay attention to people as they're trying to talk.
Okay.
So I might have to close my window so I don't see the comments, but they rephrased the question, what do you feel about putting aerosols into the atmosphere? There's a really simple answer. Sounds kind of clumsy.
Yeah.
Sounds like a stupid, clumsy human thing to do.
Oh, it is.
And that hasn't been working very well for us.
It isn't, and it's also, so once global, like climate change gets real bad in the next 10 years, the thing that inevitably people are going to do, because we're too fucking stupid to just fix the problem and deal with our carbon emissions and all these other things. So if I had to bet, if I was a betting man, the most likely thing that's going to end up happening is something along the lines of releasing enormous quantities of sulfur dioxide into the upper atmosphere, to cut down the amount of sunlight that gets through, which I feel like I saw a movie about that once. But there's...
We are overdue for a super volcano.
I was thinking The Matrix, but where they just made the sky black, because that's not quite the thing. It's albedo modifying compounds. The problem with this is sulfur dioxide works very well for this. It also recombines with all the things in the upper atmosphere to make fucking sulfuric acid. So you're basically, like, definitely making acid rain everywhere, which is worse. Or you've got the fact that sulfur dioxide is poison, so if it comes down... Who's talking about zebrafish microinjection, and how do you know that I'm trying to make a zebrafish lab? I want to know. So, like, and like, seeding clouds won't do it, because, like, you can't... There's not enough cloud. You could not cloud enough to actually deal with the sheer volume of CO2 that's in the atmosphere. So you need something that's like sulfur dioxide, which can, like, very seriously reflect infrared light, so it just doesn't get in. Whereas clouds will absorb a lot of that infrared. Like, it won't reflect it necessarily. Like, it'll take in some of that heat. But yeah, that's the thing. Sulfur dioxide gives you acid rain. So, like, chances of people or it could punch a hole in the ozone layer and then we're fucked. So, like, there's lots of ways that this could be very, very, very bad. But it's an easy stopgap to, like, very quickly drop the temperature. And humans are lazy and stupid, so that's probably going to happen, and that's very, very bad.
So, hey, do you remember our previous conversations about Project Creep?
Yeah.
What are we trying to talk about right now?
Ents. Yeah, no, I know.
Ents.
Hey, look, the half the point of
Um,
this is just have fun, just talk about whatever. So
yeah, it's true.
I'm not super worried about
Yeah.
this. I mean, I feel like
Mm,
I don't know. It seems it seems the people in the comments are enjoying it. So fuck it, let's have some fun.
yeah.
But anyway, so okay, so we talked about how the original wood paper worked, where it's like it's two.
This was the original idea.
Yeah.
And so it turns out that it's patented all the way up the wazoo. And so when I'm like, hey, I'm just going to make it myself, it turns out that it's really, really big. Like the combination of those two genes as a modification is just huge. So it's very expensive to print. And it's one of the only things that I've never had a fellow lab say, yeah, sure, we'll send you some bacteria. Like, almost like if you reach out to a scientist, they want to share. And so this was like a very unique experience where they're like, we're just not even going to respond to your email.
Yeah.
I'm like, huh, who are you guys? Okay.
Oh, the logging industry. I see.
Oh, yeah. Oh, Warehouser, hello. And so so we had to come up with some other ideas, and it turns out that there's a bunch of other techniques for modifying plants that people are playing around with them. And one of them has to do with root growth, which for any of you permaculture nerds out there, love to talk about the root depth of wheat versus the root depth of wheat now. And so there's a hack for that, like, because we basically bred that out. Well, it turns out that you can turn it back on, which is pretty cool. And the Salk Institute is actually doing work with that right now. And then the other one is a gene called SB pace. And so SB pace is like totally the Superman gene for plants.
This guy.
Yeah, is this guy. And so you don't need to, a lot of plants already have this. So we're not like adding something that didn't previously exist. We're just cranking it up a little bit. And it deals with salt tolerance. It deals with heat tolerance. It deals with cold tolerance. It deals with increase of photosynthesis, and it deals with plant size. And so basically you make Superman trees. And I'm like, cool, well, this is way easier than trying to
so
fight
for
with
clarity,
these people.
so for clarity, it's basically one gene that you can just like add to plants, and in theory, it should do all of those things. In theory,
No, not in theory. There is a lot of papers. If you type in the word SB paste, and for some reason people are liking SB paste plus tomatoes, I don't know why. You will get a whole pile of people that have been doing research about this.
yeah. I mean, SBPA is pretty neat. And the nice thing is because it's one gene, it's much easier to have synthesized. And also, I mean, it's small. It's teeny tiny. Like, I mean, if I just click on this, it is tiny,
Yeah.
teeny tiny.
And
Wait,
so
what
I
is all
can-
this? This
I
is all. I
can
mean, there's a
smack-
little extra bit here.
That's
So
the whole thing. It's like a baby.
yeah.
And so I can smack that into an agrobacterium. And then what we've been doing, what we're currently doing in the lab right now, is actually test casing this with pea plants, because shout out to Mendel, and then seeing if
I just love that. Shout out to my boy Mendel. Dab. Like what?
shout out to my boy Mendel, gonna use some pea plants. And so and we're using a novel form of genetic modification for plants where I'm basically, I deal with biology, so I'm super comfortable with reverse engineering. So I'm going to take somebody's protocol and I'm going to reverse engineer that. I'm going to see, okay, how easy can I make this? Can I make this so easy that I basically send a package of seeds and a vial of bacteria and some IKEA instructions to Justin, and then he doesn't even maybe Justin has a stroke and he doesn't understand biology anymore, and then he can just smack that shit together and um, and make it work. Because that would be cool. Wouldn't it be nice to have like some easy hands-on biology, the way that you put together that desk that mostly works? So, so that's like the end goal. And so now we're working with smaller things and simpler things, and we're trying to strip down protocols and make them more accessible, because open access to protocols, open access to science, open access to information is one of the most important things, in my humble opinion.
Yeah,
So,
no, I mean 100%.
Yeah, and so the best way to do that is to make ease of access easier. And so that's what we've been working on lately in the lab.
Very cool.
Yeah.
So, I mean, I believe I actually have this plasmid sitting in my freezer. In fact,
Yeah, you do.
In fact, I think I found a
You
second.
definitely do.
I think I found a second vial of it actually, because I think you sent it to me twice. Which, all right. So really, I mean, all I got to do,
Yeah.
like, so I just brought my my plant growth chamber home, which, by the way, I got to send you some pictures after this. I'd left some stuff in the plant growth chamber and then left it alone for a month because pandemic, and I just hadn't had a chance to pick it up from the lab. And there were a few things in there, and
Yeah.
they were like really grown,
Growing.
growing like a lot. So like there's two very sad but very much alive sterile arugula, which is look. I've got a coconut scented geranium, and I had some sterile mint going, and it went wild, like in the little plant in the little petri dish. So that's been pretty cool. Um, but
Actually, no, we're not hoarding plasmids. And in fact, the nonprofit that I started to get the cred so that I could buy materials from people that are like, we won't sell you shit. There's a loophole. There's always a loophole. And so part of the loophole is that if you make a library and then the genes are in the library, they you both simultaneously violate and also fulfill a certain requirement of a standard material transfer thing. And so I have a gene library. If you want genes, I will send you genes for just the cost of shipping. I don't like, we're not hoarding shit here. I'm giving it out for free, baby.
yeah. And so somebody asked, sterile as in can't reproduce? No, sterile as in like I grew this, like I sterilized the seeds in bleach and then grew them on sterile media in a sterile tube. So that way, if I want to culture the leaves, like if I want to take a little piece of plant and then grow it into a little like booger of plant stem cells, I can just do that immediately without having to try and sterilize a leaf that was just growing exposed to the external environment. And so this is way less stressful for the plants. It's harder to set up because not everything will germinate when you do it this way, and sometimes you got to find just the right media to grow the thing, but it saves on steps later. So it's not that they can't reproduce; they definitely can. It's aseptic would be I think a better way of describing them. Also, somebody asked about what does a plant growth chamber look like. It's literally a Home Depot bucket with like silver tape around the inside perimeter, and then some like LED grow lights. That's it.
Wait, you didn't just repurpose one of the closets in your house with some LEDs and turn
No,
it into a whole
no. I've got a whole
thing?
I've got a whole corner for that, and also like so people who've been tuning in regularly may notice that like my background is changing a little bit. So you'll notice that the new curtain, so that the bio area is in bio area. I'm going to be adding more plant. I'm going to be adding more plant growth. So there's going to be a wire rack, like a 4-foot wire rack shelf in the back corner, right behind me, with a bunch of lights on it, so I can do microgreens and just more herbs and stuff. Just because like I've got my little plant corner on the on the other side of the camera, but I need more plants. I want more plants. I need more plants. I'm huge. I'm a bio nerd. I need more plants. Also, there's
Well, I know I got a friend on Facebook who's going to send you a bunch of plants.
Yep. I'm waiting on some plants. And so I've also been buying plants from this company called I believe they're Baker Creek Seeds. The website is rareseeds.com. It is a fucking fantastic site. Like, the stuff that they carry is just really awesome. Admittedly, they're a bit low on stock at the moment because it's planting season and also a pandemic. But the stuff that they do have in stock is fantastic. I've got some peppers growing right now called habanadas, which is like a habanero but no spicy. So you get all the flavor of a habanero but none of the spicy. I also
Why would you take away all the good part?
Because the spicy is the worst part. I want the flavor, not the pain.
Okay, well, me and my Carolina Reapers, don't talk to me and my Carolina Reapers ever again.
Listen, man, I only like tasting food in one hole. I don't like tasting it on the way out. Because the only thing you can taste is spicy, and nobody needs that shit. Anyway, so I've got habanadas. I've also got some Cape gooseberries, but I'm out of room in my plant area, so I really want to have some more plant area behind me so I can have, like, my little Cape gooseberries. I'm going to get some more herbs because I'm finding that even though I've got a lot of herbs growing, I could definitely use more. Like, I don't have any tarragon, and I'd really like some tarragon. I'd like some rosemary. I'm just trying to, cover all my bases so that, say, if there's a massive food shortage because, let's say, a particular country can't pull its head out of its ass and, get functional while also not, embracing the plague. It might.
Hey, what's up, America?
Yeah, right. It might fuck with my ability to buy food. So I'm kind of in prepper mode where I'm gonna get whatever kind of greenery I can to supplement my diet of stored rice and beans. So that's the thing.
Yeah.
But yeah, so more plant area, but also it's going to mean that if now that I've got my plant growth bucket, I should actually be able to give this bit of DNA a try at some point. I'm going to have to restart.
Just call it a space bucket.
It's a space bucket. It's a space bucket. That's what it is. It's a space bucket.
Yeah, but if you Google it, it's going to look like he's growing something that isn't biology.
Yeah.
I mean, technically it's biology.
I mean, it is biology. It's just not.
Yeah.
I mean, it's not. It's legal in my country.
I mean, yeah, it's legal in your country, and actually,
it's just your country that's backwards.
Super fucking straight about this. This is one of the most genetically modified organisms on the planet.
You mean puppies?
No.
Other than puppies?
The other things. The other thing. The other thing that people can get in your country.
The electric lettuce.
Yes.
The Johnny Red Eye. What? Fuck. There's so many other funny names. I mean, just from watching too much Letterkenny, I've just picking up all the names that they've got for it. But
see, I'm 15 years older than you, and so for me, it's the Red Green Show is my Canadian feedback, and that's completely different.
Yeah, I like Letterkenny more. It's way more fun.
Well, yes.
Somebody's like, you're going to want to liquidate your assets as well. I recommend silver or toilet paper as your value storage medium.
Okay, so no, we're not going to. No,
let's not get into this because
okay. So can we get back to biology?
Yeah.
Okay.
Yeah, the devil's lettuce, electric lettuce, laser lettuce, all fun names for the same thing. So yeah, space buckets are great tools. Just most people use them for not genetic engineering of single genes at a time.
Is it for a very hyper-specific type of genetic engineering?
Yeah, mostly done via breeding. Because for another plant that I wish that people would actually genetically modify instead of this slow breeding bullshit.
Wait, pause.
Yep.
Pause. So Dr. Jackal, hello. I have noticed you in the comments. You were making good comments, and I'm going to put a pin in that, and I will start working on that with Justin. That sound good?
There's a link to a GitHub repo full of plasmids under the video right now. I can upload these.
Right, but you're also.
I can upload these can I upload these plasmids to that?
Yeah.
All right, cool. Well then, so yeah, to
Do
anyone
it.
wondering, any of the
Do it.
plasmids
Yeah.
that we talk about on the show end up on the GitHub repo for Whose Gene Is It Anyway? Because that's the show where we actually do a lot of genetic design. But I'll make a folder with
Yeah. I'm.
for bees, and I'll make a folder for ents. So anyone who wants to download the thing,
Yep.
they'll be available there.
Any of the papers, any of the .dna files, anything you want.
Uh, the mostly DNA files. I usually put links to papers, but yeah, there's stuff will be
Whatever.
there.
If you're making a folder, just throw some PDFs in it, dude.
Yeah. Anyway, so that stuff is all going to be available. Just check under the description of the video. There's a link to that. Yeah. So yes, all those things are available. And if you'd like to learn how to like design these pieces of DNA or work with these pieces of DNA, there are live streams for that thing. Like the Whose Gene Is It Anyway live streams. We go through the research and design process for a piece of DNA like this. For example, let me see if I can just pull one up really quick for one of the ones that we did previously. Ooh, actually, this is a good moment to show the milk and eggs plasmid again. So the very first stream we did, we designed two plasmids. One will make sort of milk, and the other one will make egg white, produced in yeast. And so this was here's the first one. So this is what we actually designed on the stream. And so all of these genes are the different proteins that come from milk. And if you want to see how we designed this and how this works, go watch that stream. And eventually, we did a fundraiser. So the order has been put in for this plasmid. Although before I put the order in, I actually made some modifications. So this is the thing I actually put the order in for. So I basically took the useful part that we designed on the stream, so from here to about here, and I literally like just copied and pasted it and stuck it into this plasmid, which is an integrating plasmid. So that way, basically, once the way this works is you put this into yeast, and it's not labeled on the map, but there's a little spot right around here and a little spot right around here. And you basically cut this piece of DNA before you put it into the yeast, so it's a linear fragment that starts like about here and ends right about here. And when you transform this into the yeast recognize that there's a broken piece of DNA, and so it'll actually match the bit that's here and the bit that's here to the yeast genome and integrate this into the yeast genome so it's permanent. So this way it'll continue to make this thing even without selection, which is important if you want to be able to drink this. So this is the thing that got ordered. And yeah, so if you want to learn how we design this and like how all these different parts work and like what is a P2 or a T2A, like what is that? How does that work? I would highly suggest watching those streams, and then you can actually like learn the thing. And all of it's open source. We did a fundraiser to have these ordered. So once they are ordered and I know that they work, then I'll be releasing them. I'll be posting them I'll be submitting them to AdGene to start with, because that's easy. And then anyone who wants them can just order them. But then in theory, I may end up making a store on my own site, but I haven't decided if I want to do that yet. But yeah, so there's that's deer milk. And I don't know if I mentioned, but the milk proteins that we used came out of deer because that was one of the only animals that weren't patented.
Okay, but so tell me this, Justin. Are you going to get yourself a deer and milk the deer so that you can compare your product? We need to have second-party verification. This requires getting.
I don't care if it tastes like deer milk. I just needed the proteins. I just needed proteins that weren't patented. It should just make milk. Period. I checked the difference between deer milk and cow milk. Is mostly a ratio of the proteins and some very, very tiny modifications to the amino acid sequence. So I chose deer specifically both because it's a sequenced animal and also because it's very similar to cow milk, so that there shouldn't be any noticeable difference. And it shouldn't taste different.
No. I mean, this makes me feel. I'm thinking about the kind of the battle between the rabbit industry and the chicken industry. which are both considered white meat. And so there was actually this whole thing where, you didn't used to get, you could choose, but they were trying to domineer. And so they were like, chicken season, no rabbit season, no chicken season, duck season. And so chickens won out, but like nobody eats rabbit anymore.
Which, I mean, to be
rabbit
fair,
tastes so fundamentally different than
it's
chicken.
not good.
At this point.
And also, I mean, it's that.
It's you just haven't had it prepared properly.
No, I have. That's what I was about to say. You have to prepare it properly. Whereas chicken, you can fuck up royally and it still tastes fine. Like, I mean,
it's the most basic bitch of meats.
It is. You gotta fuck it up real bad before chicken doesn't taste good. Whereas rabbit is like, if you so much as like sear it for a second too long, it's like, this is like eating sand. Like, it's just not good.
Don't get good, dude.
Hey, no, it's not a problem for me, man. It's like, go to a restaurant, order rabbit, be very disappointed when it tastes like eating sand ravioli.
Yeah. So, I mean, my point about bringing this topic up is, what if deer milk is just like, oh fuck, why?
Well, look, I also chose it because we needed something to start from that wasn't patented, and like all the usual things that makes, like, in the patent, and this was what was really, really frustrating in the patent where they're like making it was the Mufri patent, like that was where I got this from. It was in the Mufri patent. Not only had they patented all of the things that made sense, so like goats and cows, the list of animals that they included on, like, we claim these, fuck you, you're not allowed to use them. It was like every mammal they could think of. Fucking like rat, human, gorilla, orangutan, fucking woolly mammoth. Like, they really tried to just be ridiculous. It was absolutely ridiculous. And I'm like looking at this list going, no, horse is included. No, that's no good. Rhino is included. No, that's no good. Then I was like, wait a second. How did they miss deer? Like, it's deer. Like, you're thinking of woolly mammoth, but you missed deer? So yeah, that was where that came from. Like, I had to find something that wasn't on the list, and like during the stream, like people suggested other things.
See, now that makes sense. That makes sense.
That was the reason.
I can get behind.
That was the reason. It's so I can basically copy left milk yeast. Is the reason I chose deer, because it's not covered in any of the patents, and so it's like I can submit it to AdGene, and now all of you can go fuck yourselves with your stupid patents. Like, anyway,
I mean, that's what we do.
Yeah. Somebody said, "What's the danger of prion production in working with animal proteins?" Well, unless you're working with prion protein, you're fine. don't work with prion protein.
The whole point of using laboratory materials is that they're all purified and checked for this stuff.
Also,
so like when you get your growth media,
prions don't just happen from like random proteins. It's like one protein. Like, it's a specific protein is prion protein, and like,
well,
I mean, there's
a lot of different types.
There's a couple of them, but like,
protein.
It's like a small handful of proteins can become prions. Like, it's not all of them.
Yeah.
So yeah, that's the thing. So it says, will you be sharing the plasmids for your other projects to GitHub? Yes, they will be posted on GitHub after this goes up. I really should add all the spider silk ones. You know, I think after this stream, I'm gonna go through and upload a lot of the plasmids that I've designed to GitHub, because I'm really trying to lean into the like everything is open source type thing. The lactase, the new lactase plasmid will be up there soon. The spider silk thing will be up there soon.
Somebody has asked a legal question, and I feel like I definitely need to answer.
Okay.
No, you don't. You just need to have a business license.
Sorry, just to clarify here. Do you need a license to buy prescription antibiotics for genetic engineering? If so, how do you buy it without having a degree in medicine? So yeah, to follow up on what Gabe said, no, you don't.
So you start an LLC.
Yeah.
That's profit. That's it.
Yeah, basically, like, as long as you like, Thermo Fisher doesn't care what your degree is. Like, this is the thing that when you're in academia, they don't tell you. They don't give a fuck what your degree is. They just want to see that, do you have a business, like a business license, and a commercial address. If you have those two things, they will sell you whatever you want, regardless of what your qualifications are. So that's just how you do. So if you want to do biology, really, you should set up a business immediately and find a commercial address you can ship to. Is the biggest thing. Quartz turtle. Did I not? Oh shit! I suppose I forgot to upload the glass turtle plasmid. That's my bad. Like I said, after this stream, I'm gonna probably spend half an hour and just like upload all of the new weird plasmids that we've made. And I'll make a folder for Gabe's stuff, and I'll make a folder for my stuff, and it'll be great. So yeah, basically,
so much sharing.
Yeah, it's all sharing all the time. Someone says that's a bit terrifying, but also great. I mean, that's the way it should be. Like, you shouldn't need a license to do fucking biology. Like, if you need a license.
Okay, so this is the thing. But also, they're terrifying, but it's also great. It's like, can I order bomb ingredients? It's like, yes, but if you are good enough to make a bomb, you also know that you're going to blow yourself up.
Yeah.
And remember how we talked about how biology doesn't care about us?
Yeah, there's a reason why, there's a reason no biohacker is going to make a virus. Like,
yeah, it's just not a thing.
It's not a thing
because the first person who's going to get hurt is you.
Yeah, no biohacker would ever make a virus because no biohacker is stupid enough to try. Like, because unless you have a BS4 lab, you're going to fucking die. It's not a good plan. Like, it's just the way it is. Like, people don't mess. And also, like, there are actually, and like, outside of just being able to buy things, there are actually laws in various countries about what you're allowed to genetically engineer. So, like, in Canada, for example, you're allowed to do basically whatever you want with 3 main exceptions. Exception number 1, no working on animals without a license. Like, you actually have to, like, do paperwork to be able to work on animals.
Yeah, you definitely. Animal testing is sketch.
Yeah, any animal testing is sketch, and they will throw the book at you for it. No toxins. So
yeah,
it's
actually way easier to experiment on myself than it is to experiment on a mouse.
Yes. So like, you're totally allowed to use Black Widow genes if it's the silk. If it's the toxin, they will throw the fucking book at you if you attempt to grow any of that. So yeah, no toxins, no animal work, and if it's food, to be able to sell it, you have to go through an enormous amount of paperwork, and get it like qualified. But I mean, there's lots and lots of companies that are doing some really amazing genetic engineering for like food stuff. I mean, somebody, like, inevitably in every single stream, somebody's like, what if we make THC yeast? THC yeast exists. My friend, a friend of mine who I'm trying to get on as a guest for one of these live streams, has a company that has successfully made THC and yeast, and they've got millions of dollars in investment and are like spinning it up to be able to literally produce tanks of the stuff. So yeah, it's totally a thing. Like, there's lots of great things you can do, but you also have to be aware that there are some laws and some lines that you don't fucking cross. Infectious agents and toxins are super, super illegal, so you don't touch them. This is why I work with Silk, because I just want to make weird, goopy yeast. I don't want to fucking make anything dangerous because I'm not a dumbass. Like, I want goopy silk bricks made of yeast. I don't want poison. But I mean, as long as you're not buying, toxins or infectious agents, you can buy whatever you want. Nobody cares. Like, I've been on the phone with Health Canada before, and they're like, "Why are you ordering this?" I'm like, "It's just DNA. What's in it?" I was on a call with Health Canada when I was ordering some DNA, and it got stopped, and they wanted to know what was in it. They're like, "Is it infectious or a toxin?" And I'm like, "No, it's just an empty yeast backbone." They're like, "Okay, that's fine, cool." They even explicitly said, they're like, we don't regulate DNA, caveat being, unless it contains toxin genes or infectious genes. At which point, then yeah, don't fucking do it.
Cool. So are we we talked about all the stuff. Are we gonna answer some more questions?
Yeah, I mean, that's basically what we're doing at this point. We're shooting the shit and answering questions. Because I mean, we've talked about the projects. The Ents thing is dope as shit. And hopefully I'd really like to, I want to do a video on it at some point. I have so many, project creep. I have so many other things going on.
Artem!
Oh,
Sorry.
there's a wild Artem. Sorry, he's another friend of ours. And so yeah, so some more questions. It says, Gabriel, do you have a quote unquote proper degree in bio?
Yes, I do. I have a degree in molecular biology, and I worked for 4 years in an chemical engineering lab. And I also worked for 2 years in chemical engineering industry. So I know the shits,
He
but
is
they're not necessarily necessary.
yeah. That's the thing. He's like a proper professional, whereas I'm a hack. But you know, he has a degree. I don't. Just because I also get that question all the time. They're like, "What's your degree in?" I don't have one. I tried, but I got too bored, and they wouldn't let me
Justin has a degree in being really great at YouTube.
Yeah, basic. I have a degree from YouTube University. No. Yeah. Then I'm seeing some comments about like what about in Europe? No, you don't do genetic engineering in Europe. It doesn't. It's not a thing. Like they want you to have a license to like work with GFP because they're idiots.
Okay, so
I when it comes to like anything legal wise in the EU or just Europe or the UK, it's just ass backwards. Like they just tried to. I think they're pushing legislation through that now even like mutagenesis made plants count as genetically modified, which personally I find this awesome because it's like if you're going to be obnoxious about one thing, you might as well be consistent. Um, but it's also a ridiculous, like absolutely ridiculous classification, and like people get so bent out of shape. But also, the regulators are a million years old and don't know their ass from a hole in the ground. So like, this is to be expected. Why do you need an LLC for just lab equipment? Because sometimes,
so when big business and large corporations and the government get together and they're very much in love and they're ready to have a baby, they don't want anyone to touch the fucking baby.
Basically, like unless you're buying it off of eBay, like if you want to buy a piece of equipment from Thermo Fisher, like they'd be like, okay, cool, we're gonna need your business license and commercial address, please, because why would a residential address ever want to buy very expensive bio equipment? And then there's like us being humongous nerds being like, because I just want to make nice plants. Like it's just the way it is. How hard would it be to make artificial megafauna? Yes. Very.
Yeah, the biggest problem has to do with the fact that the genes are inside. So like our best DNA work is done with eggs.
Yeah.
And so bacteria are great because they don't even have eggs. They're just like little jelly bags, right? And so we can just be like, hey, I'm going to yeet some freaking plasmids into you, and you're just going to make them until you don't want to anymore. And then we're pretty good with fish because their eggs are on the outside. We're pretty good with chicken because their eggs are on the outside. We're good with plants because it's a whole nother thing. They don't have eggs. But they're on the outside. But like when you start talking about macro species, and for me,
like
macro
animals
species,
specifically.
Yeah, anything with a spine, basically.
Yeah.
Like anything with a spinal column.
And not eggs.
They keep their and not eggs. So I guess technically platypus are macro species and they make eggs, but they're an outlier event. Let's just move past that.
They're also the only species that can make their own custard.
They're also the only earth growth factor and has like no more platypus. So
this is so weird.
So it's very hard to modify things that have spines. And all of the macro species that exist or have existed in the past 10 or 30,000 years have spines, and they have the babies on the inside. And so modifying them is a lot more difficult.
Yeah. Yeah, like and also trying to make an entire species is like it's so many genes. Like all the things we've been talking about are like messing with like one gene. Like think about the spider silk. The spider silk thing has been literally years of frustration and hardship trying to get the stupid thing to work, and that's one gene. Like trying to engineer an entire species, then you're messing with like Hawk's genes, and as soon as you start messing with that, everything is out the window. Like
Yeah.
that's like you change
You know,
one letter and you grow
the
a
first
second
rule of
fucking
gene
dick. It's
editing.
not good.
Yeah.
Yeah,
You get like literally arms growing out of eyeballs.
yeah, it's not good. You don't mess with Hawk's genes if you don't know what you're doing, and nobody knows what they're doing, so you don't mess with Hawk's genes. um,
Yeah.
like it's just not good. Like, you know, making like a snake out of a fish, I suppose you could do it, except we know so little about like. But I mean, think of it this way: both me and Gabe are professional genetic engineers. We do genetic engineering all goddamn day. But the idea of making one species do something weird, like both of us go like we don't know shit. Like we don't know nearly enough about like all the intricacies of DNA and regulatory stuff and all the weird ways that DNA can come together to make organisms do things. Like we just don't know we just don't know. We don't know nearly enough. Like it's just not viable for probably the next 25 to 50 years. Like is how long it's going to take to understand all this shit. And even then that's for like a species. Like you could spend your entire life from today forward studying fish in order to make a fish snake. And it'd probably take you 20 years to figure out which gene you actually have to kick just to get it to make a longer spine. Like, it's not easy.
Yeah. So I want to just say really quickly, because I feel like we're kind of wrapping up, that anyone who has any questions about this type of thing, you're more than welcome to find me online. I'm easy to Google. I exist on Facebook, or you're on YouTube. Yep. And you can DM me. I have an infinite amount of patience, and so I will. Every single person who had a question that flowed through. Yeah, I know. Justin's like, I would never do this.
I would never do
like,
this.
Oh god.
I get so many emails that I can't keep up with. Like you're brave.
Yeah.
You're braver
Anybody
than
who has
me,
a question,
friend.
anyone who has a question, you can DM me or email me or Facebook message me or whatever the fuck your social media platform is, and I will answer it.
Everybody just message him on TikTok. Just do it.
Oh god, hey, I got a really cute TikTok channel. Okay.
Okay, well, so for those who are interested in your stuff and the things that you do, other than just DMing you like a fucking savage, like where can they find your stuff? Like where do you post the things that you work on? Where can they find your stuff?
This is an interesting question, because I have only just recently started posting little baby videos on TikTok, and all you got to do is Google my name. I don't actually post about my work because I feel too much media hype about pushing things to be successful or to be a product. And so I just don't do that. Because I also have a fair amount of media exposure, but in a different way than Justin does. And so I only ask for it in certain ways. I would rather connect independently. So you can't, if you look at me on Facebook about food, if you find me on Insta, it's going to be shit about food. Like if you find me on TikTok, there's going to be some cute sell videos. And if you want to know more about my work and not have it distorted by media exposure, you're going to have to talk to me in person.
Yeah, basically. Like Gabe is a big like find him at a conference and like buy him a drink and then, you know, talk his ear off kind of thing. But in case you're living under a rock, I don't see any conferences happening for the next 2 years. So, you know.
Yeah.
Man, I was so excited to go to VidCon this year.
You're gonna find my work. You're gonna find my work in Justin's GitHub.
Yeah, basically. All the plasmids that we've talked about will be in the GitHub. And yeah, if there's, I mean, I'll put some links to Gabe's various channels in the description. I didn't do that in advance because I'm a turnip and was, struggling to make sure the stream was working properly. But yeah, so there'll be some links there. I mean, we tend to like, when there isn't a fucking pandemic on, like, we tend to show up to the usual like bio conferences, so that's always a good time to find Gabe. You'll usually find him in the hallway with a drink, talking with someone interesting. It's just how he rolls. So yeah, that's the thing.
But also, let me just lean into this.
Oh God.
Post quarantine.
Jesus.
We eventually have to be post quarantine.
In 2 years.
Justin, how did we get to meet each other so much? How long did you live with me?
Oh God. I mean, it was more than a year, I think. Like
it
was more than a year in multiple countries. Like,
yeah. So if you want to learn something, you can just tell me that you've decided to come to my house.
Oh God.
And I'll say, okay, dude. Post quarantine. Post quarantine.
Yeah,
stay home, stay safe.
Yeah, Gabe is pretty good about the like teaching people in person thing. You just need to be aware that like he will explain why you're stupid in like in real time.
Hey, I'll explain why they're smart too. Well, whose fault is that, Justin? Okay, how do you feel about that, and whose fault is that?
Yeah.
You walked into that thing.
Yeah. I mean, I feel like this is the sort of like entertaining shit that like you don't see in the videos. I mean, one of the reasons I've liked doing these streams is, I mean, I get to kind of like actually let my actual personality out a little bit instead of the like very sterile version of me that you see in videos. But like if you think about like the Neuron video or the Meatberry video, like all just the ridiculous shit that me and Gabe would get into, just like just doesn't come through because like all the audio is always muted, so it's just the two of us being like turnips while we're like, I don't know what's going on.
Stingray Squad. I've had up to 12 people in my house before.
Oh God,
so
yeah, that was a fucking thing.
And remember Chile?
Hush, we don't talk about that. Didn't happen. Someone would be like, you're about to be the leader of a commune. See, you joke, but he'd be into that.
I wouldn't be into it because intentional communities are fundamentally flawed.
They are, but
that's a whole nother conversation. We're going to talk about biology. We're not going to talk about how libertarians.
Yeah. But anyway, somebody I was curious how the ultrasonic booze actually was. I mean, you saw our reactions in the video.
It was great.
It was fantastic. I have some. I like tucked away on a shelf. I made more. Like, I'm seeing some stuff. Do you think it'd be possible to modify nerve cells to produce light when they get a signal?
I would definitely do the cerebral Licin again.
Yeah, so I mean, cerebral Licin's great. I took the video down because it was too sketch for YouTube, and I didn't want to risk a channel strike because this is literally my job, and also, the channel just passed 500,000 subscribers, which I realize we're 2 hours into the stream and I haven't talked about this yet, but like, I'm fucking thrilled. Like I'm super grateful that like, people, I can't believe there's 500,000 people who watch the things that I make. Like, it's craziness. I'm like so excited. It's amazing. What else we got? Yeah, the 500K is pretty cool. I'm hoping that we hit 1 million before the end of the year. I want that gold play button so goddamn bad. Like, so bad. So hard to know what things are going to do really well and what things are just going to be like ass garbage analytics. It's But so I'm just trying to lean into like what are doing the projects that I think I'll enjoy the most, and like I think will be the most interesting. Like, I mean, it's what,
Dude,
yeah.
no, seriously, we should be hosting on Pornhub.
No.
Like, what the fuck have we been doing? Like, YouTube is letting you down, and I'm here dicking around on TikTok,
Yeah,
and
just
we
streaming on
could be
Pornhub.
making science porn. Yeah, science porn.
I feel like I don't know. People, it's not a good plan. I'm not into it. I'm
We
so
need to
not
let
into it.
apps, okay? We have been doing this for hours,
Yeah.
and I think we've actually covered all of our topics.
Yeah, we have. I mean, so let's
Okay.
take another like 2 or 3 minutes just to see if there's any questions left, and then I think we'll wrap it up for the call, or wrap up the stream. I think we've got a couple things left to chat about once this is done. Um,
Mm-hmm.
but yeah, so if there are any questions, if there's any questions that you want to ask about Gabe's projects or my projects or whatever else, now is the time.
Yamageddon.
Yeah,
Short answer is no. Meat berries. Kind of didn't work.
Wait, what? I mean, it worked. Oh, did I tell you somebody messaged me on Instagram and they're like, I recreated the meat berry. Yeah, somebody actually, and they did it with
nice.
and they did it with GFP modified cells, so you could fucking see them. It was great. Like, I was so thrilled. Okay.
Thank you for all of your time. I don't really know how to. I don't do this, so
Yeah,
yeah.
I think this was fun.
Cool, guys.
So the next stream is going to be on Monday. In theory, my new video capture
The
card.
P. Stypticus we don't talk about blood fungus.
It wasn't Stypticus. it was Microspora.
Okay, that was the P. microspora.
Yeah,
Sorry,
it's
you're right.
it eats plastic, but also makes.
Hey, I was triggered.
Yeah. No, blood fungus
Yeah.
is it eats plastic, but also produces toxins. And oh, by the way, it can totally live in blood. So we stopped working with that because we're like, yeah, pathogenic plastic eating blood fungus sounds like a bad plan.
Started working with it again.
Well, yeah.
Will you ever find love in a petri dish? No, but you might find love with someone who's also looking at the petri dish.
Yeah, always.
Never did that night vision eye drops again. You know, it's great optics. Ha, that's a pun. But nobody actually gives enough fucks to fund any further research on it. But I do have some friends and associates who have done it individually and done things like gone out stargazing or their own self-experimentation.
Yeah.
So
with the night vision eye drops, since we're there, and for those who aren't aware,
and blood fungus.
Okay, well, so for those who aren't aware, Gabe is the night vision eye drops guy. So if you look up night vision eye drops, you'll see this fantastic picture of him with black eyeballs, which I love using in videos because I know it drives him insane.
Never gonna fucking escape that.
Never, ever. Like, as soon as that picture happened, you're like, yep, this is my life now. But so one thing that I did, I've always been curious, or not always, but for a long time I've been curious about that particular project, is rather than using C6, which just makes your eyes more sensitive kind of generally, is using a different dye molecule and seeing if you can basically get tetrachromacy using like tetrachromacy eye drops, just because I think that would be dope. I'm just also not crazy enough to put fucking insulin in my eyes to test it. So,
it was a completely well-researched amount of insulin.
Hey, no, I'm not saying it wasn't well-researched. I personally am just not of the put insulin in your eyeballs clan, you know? But tetrachromacy eye drops sound really cool. It's just a shame that I have a bazillion other projects and no time to mess with that. Somebody, like, I'm seeing tons of like COVID testing. I am literally working on a video about COVID testing. If you look on Instagram, there's some of the first results from it. We've just ordered. I have 3 different COVID tests that I'm going to be showing on camera. So like, it's happening. Fucking chill. Like, somebody says just because it's dope is the best way to do science. Yeah, that's the reason I do anything. What are we looking at? What is that?
I mean, that's the remainder of the plasmid.
Oh, okay. Cool.
Yeah. Yeah, just because it's dope. Yeah, actually, I don't know. Have you realized that when you're interacting with reality and you do stuff and it's not dope, that it really sucks? Have you considered just only doing dope stuff? And this doesn't mean, Karen, that you're going to sit in the bathtub and drink a bottle of wine and eat another pint of Ben and Jerry's, but you should maybe consider not being miserable doing what you do.
Yep. I saw one thing recently, basically talking about, like, you know, if people are making more from unemployment than their job, like, are you worried that providing all this new unemployment would prevent them from coming back to work? And I can't remember, I think it was a senator's response was basically like, if that's something you're worried about, you should be paying your workers more. Like, the answer to this is you suck, be better. But yeah, like doing, like, especially with science stuff, like something that you're going to be spending your years on, like, and your own blood, sweat, and tears, ideally not in your petri dishes, but just separately.
Hey, no, some people have done some great projects with that.
Yeah, that's true. You know, if you're going to do something in science, it's going to suck the entire way through, so you might as well enjoy it. Like, it's going to be hard and you're going to hate it the entire way through. Like, I love the Silk project in theory. I'm very excited for it, but there is not a day that I don't wake up hating the shit out of it because of how hard it is and that it's not working. So, like, if you're going to pick a science project, pick one that you can just, like, love enough that it overtakes the amount that you hate doing it. Because that's just, like, science is just a big exercise in, like, embrace the suck. And I've talked about this in, I think, every single stream so far.
Super true story.
It's true. You just embrace the suck and find something you don't hate. Like trees or bees or silk or whatever the thing it is that you love doing.
Yeah.
Basically. So yeah, that's the thing.
I feel like that's a good punctuation mark.
Yeah, I mean, it is. It's okay. Let's do one more, and then let's wrap it up for the day because it's already been 2.5 hours. Would using GMOs and hydroponics be useful to increase the yield? I mean, that sure. Okay, that one was kind of short.
Yeah,
so
that's a simple one.
Yeah. Somebody's like, okay, how about a contact lens that augments the amount of light received? Okay, so this is actually really cool because this is something I've been reading up on. There's a series of papers. So if you've heard of the Enchroma glasses that, like, fix vision for colorblind people, you can totally do this with not colorblind people. So if you basically force a division in the optical spectrum, to make your eye, like, to make it easier for your eyes to tell the difference between colors, you can essentially end up with, like, virtual cones in a sense. So, like, you get way more color perception because you're able to tell colors apart, which is basically what tetrachromacy is. So they in the paper they were doing this with all, like it was glasses, not contacts, but they were able to add a virtual cone to non-colorblind people, and they did the math. I think that you could add, I don't know if it was 4 or 6 extra, like, virtual cones just by, like, layering the materials right and blocking out just the right wavelengths. you could basically not get tetrachromacy, you could get like octachromacy, just using like special glasses, which I thought was just so damn cool. I don't know if you can make that into a contact lens format, but yeah, that's pretty wild. Do you have any links? Not handy. I don't. I'll dig up the paper. I'll make a note. I'll dig up the paper after the stream, and I'll post it in the description because it's pretty cool.
We're just gonna have to make a folder for everyone.
Yeah, basically. Like I said, I'll do links in the description to everything that we've talked about.
Wow, you're gonna have so much fun.
I know. It's always a solid like half an hour of just like going through everything. I'll rewatch streams just to see if I missed something I was supposed to like put in, which is painful. Okay, cool. So that's we've been saying 5 more minutes and 5 more minutes. So we're gonna wrap it up.
5 more minutes. 5 more minutes. Yeah, we just like hanging out with each other. That's what's going on right now.
Basically, and also the comments are entertaining. But okay,
yeah. Now we've hit the point where people are talking about glow in the dark coffee. So
yeah, so that's that's I think a good stopping point. This was a lot of fun. We may have I might have Gabe on another future stream at some point. I'm gonna be doing more streams with guests because this has been a lot of fun. I've got someone from the Open Insulin Project I'm trying to figure out a time to have them on. I've got a bunch of really cool bio friends who I'm going to have on as guests at some point. So the streams, as always, are going to be Mondays and Fridays at about 2 p.m. Eastern. The next stream will probably be either a Whose Gene Is It Anyway, or maybe we'll be doing some lab work if my capture card has arrived. So we're going to, be sure to tune in Mondays and Fridays. The stream will be in the same place. Same time, same bat time, same bat channel, all that stuff. Links are in the description. Thank you very much.
Are you even allowed to say that?
What? Yeah. Yes. It's my stream. I can say whatever the fuck I want.
All right.
But yeah. So yeah, I'll have Gabe on again, and like we said, all the plasmids that we talked about will be in the GitHub. Link to the GitHub will be below. If you're looking for if you're interested in how my projects are coming along, just go to Instagram. There's links in the description. It's where I'm constantly posting stuff. So if you're wondering when is the next video going to be, just go to Instagram. You'll see the things I'm working on. It's where it is. But yeah, other than that, I hope everyone's staying safe and happy and well, as well as you can be under the current situation. Don't lick doorknobs. Have a good day. Stay healthy. Remember to exercise, eat your fruit and vegetables, and we'll see you next time. See ya.
Peace out, Sailor Scout.